Hi everyone. So first of all, thank you so much for giving me this Best Researcher Award. I would like to thank the International Research Awards for New Science Innovations. And today I'm going to present a topic which is also a part of my PhD thesis. It says protein structure and stability modulation due to macromolecular crowding using biophysical techniques and NMR spectroscopy. So I was doing my PhD under the guidance of Professor Ramkrishna Vatrevu. So, as all of us know, that the proteins so far have been studied only under dilute buffer conditions. So, whatever we see right now over here, so this is an energy landscape of a protein that has always been characterized only in dilute buffer conditions. But within a cell, the condition is not the same. There are many other things that are present within the cells, like cell organelles, other macromolecules, and so many other things due to which the entire the entire area is completely crowded, and because of which the protein may or may not behave in the same manner that we know it is behaving in an in vitro condition. So that is the exact topic of my research, where I'm trying to find out how the proteins will behave when there is a lot of other molecules which are not participating directly in the reaction are present, and what will be the energy landscape in such a situation. So right now, just for an analogy, you can see if you are alone somewhere, you tend to be you tend to keep to yourself, and when you behave in that way, and where what you do and the way you behave is only known to you, which is well characterized by you. But what happens when you're present within a crowd? Definitely you will not behave the same way that you behave when you are alone. So that is what we are trying to find out, what proteins will do when this thing will when this is a scenario within the cell. So inside the cell there are a lot of elements, like I already told, and because of which this entire scenario is also called as macromolecular crowding. So macromolecular crowding can manifest its effects in two specific ways. One is called as excluded volume effect, that is when there are a lot of other molecules which are present. Because of their presence, the protein does not have the ability to function or exist within the cell. So the space is constrained. Because of that constrained space, it is forced to take a smaller shape or a smaller fold than what it generally does in an in vitro environment. And the second are soft interactions. So soft interactions are basically when the protein will non-specifically interact with the n number of molecules that are present around it. Now, because of both the scenarios, you can either have a stabilizing effect or a destabilizing effect. It is generally seen from the literature that exerted volume effect always stabilized the protein, because it was making it to stay in a compact state, and to stay in the compact state is the nature of the protein, which is governed by the thermodynamics of the protein. When it comes to soft interactions can be stabilizing or destabilizing. So stabilizing when when the forces are good, when it adds up to protein stability, they are stabilizing. When it does not, when the interactions are at some other place where it is distorting the protein structure, then you see destabilizing effects. So this is all in all a small overview of what macromolecular crowding can do to a particular protein structure. So now you need a very good test protein, and in my case, I have taken chemotaxis protein Y, which is completely thoroughly analyzed in vitro conditions, and it can be viewed via multiple eyes. So the structure of this protein can be seen via fluorescence, NMR spectroscopy, CD spectroscopy, and everything. And that is what I'm exactly going to discuss in the upcoming slide. How do we study the structure and stability of the protein? So first you take a protein, you can do circular dichroism, you can do a far UV CD spectrum for its secondary structure changes, you can do a near UV CD spectrum for its tertiary packing environment. Then you can do fluorescence. Under fluorescence, you will have intrinsic fluorescence, extrinsic fluorescence, and time correlated fluorescence. Intrinsic fluorescence can be given by this particular protein of choice, that is chemotaxis protein Y, because it has got a single tryptophan. So it will give you an emission maxima at around 345 nanometers or so. Extrinsic fluorescence probes like ANS can be used. This particular protein has got a buried hydrophobic surface area, which when opened, ANS goes and binds to it and shows an increase in the extrinsic increase in its fluorescence. Time correlated fluorescence will tell us how many conformers this particular tryptophan residue is attaining in its dynamic state. So characterizing that will help us know actually what is the population of the tryptophans that are exposed or buried, or which are in the native state or in the non-native state. Then we have a backbone HSQC spectrum available for chemotaxis protein Y, which can be used as the fingerprint spectra. And when you add Crowders to it, you can actually see what is happening at a residue level. So that's an added advantage. Then we can do thermal denaturation experiments where we can do denaturation in dilute buffer and in the presence of different macromolecular Crowders and see whether it is increasing or decreasing. Then there is always an option of molecular dynamic simulations, which can be used to understand things in a better manner. So that's about how we are going to study all of this in by using different techniques. So like I told you, chemotaxis is a chemotaxis protein Y is a 14, approximately 14 kilodalton protein. It has got a single tryptophan residue and it interacts with multiple partners as shown in this particular figure. So it can interact with the FLIM protein that is towards the motor end of the motor end of the bacteria, and it also interacts with key A and key Z, which are a part of the receptor complex that is present on the other end of the bacteria. So this is just some biology that we need to understand. So Kiva has got multiple interacting partners. That is what we are supposed to understand from here. Then, one very important thing is, what is the effect of crowding on the structure and stability of Kiva? That's the first question that we have and we can explore. Second, what is the effect of crowding on magnesium bound state of Kiva? Now, why does it become important? Because this particular protein displays two distinctive conformational forms, that is the apoform and the holoform, and they are structurally different. So it becomes very important to discuss what is happening in the presence of magnesium and in the absence of magnesium. So this small illustration over here tells you what is the difference between the residues and the orientation of the residues when there is magnesium bound to it. So first we purified the protein, of course. So we have a PET21A vector, and then from there we do size exclusion chromatography, we do IPG overexpression, we do a size exclusion chromatography, and we obtain a single band protein, which is more than 95% pure protein. To this particular protein, I have also done a mass spec and I have found out the mass of this particular protein, which comes very close to the theoretical mass, that is about 13966, and I'm getting 13965. So the purity of the protein has been assessed. We know that we are working with Kiwi. So moving forward, now we also did a complete 3D uh, 3D backbone assignments for each and every single NMR assignment peak, for each and every NMR peak that we get. So this spectra, whatever you see over here, is an HSQC spectrum recorded on a 700 megahertz spectrophotometer by using a different array of triple resonance experiments. So the way it works is that we try to get as much information as possible from a triple labeled protein and try to assign each and every peak to one specific amino acid. So identifying, so these are some of the experiments that I have done for assigning the backbones of each and every NMR peak over there. HNCO, HNCACO, HNCOCA, these are all standard set of triple resonance experiments. As you can see, in all these things you get information about different nuclei or different position of the nuclei within the protein sequence, and from there you can actually assign each and every peak of this particular protein to one specific amino acid, identify the spin system of that particular amino acid, and then attribute that spin system, identify the amino acid, and then give it a number based on the sequence that we already know. So this was a thorough exercise that was already done, and this is one of the strip scope that can be viewed in Kara, which is computer aided resonance assignments. It's a software that is already available. So as you can see, this is a standard CBCS strip scope, where you can see that there is an information about the C alpha of that particular peak, C alpha minus 1 of that particular peak, and C beta minus 1 of that particular peak. So if you go by this trace, so you can easily figure out, if you can see, so here it is given as C beta minus 1. So this peak corresponds to the peak previous one, which is the C beta of this peak. So this is a stretch of amino acids where you can actually generate information. And like I told you, this is a strip scope of CBCA NH that was shown to you right now. So this is a part of the NMR assignments that I have picked. So you can see C beta minus 1 is 40.337, which is again going back and pointing to 40.4. So that is the approximate we are trying to match, right? So, and then there are certain specific amino acids that you can pick and see and try to start assigning from there. So alanine is one such peak where the C beta comes in a very very high range. Serine and threonine is one another specific peak in which if you load the CBCA NH strip, you will see that the C beta of this particular residue will be below the C alphas of those residues. And then glycine does not have a beta of itself. So whatever beta loads up in that particular strip scope, that beta will be of -1. So these are some of the identifiers where you can try to do it. So now, what happens when you try to what happens when you try to study the structure and stability of this particular protein in the presence of PEG 20,000 and FeCol 70? So very quickly, I'm going to tell you that when we subjected this to a far UV CT spectra in the presence of PEG 20,000, we saw that the secondary structure of the PEG reduced in a secondary structure of KY reduced in a concentration dependent manner. The same was also observed in the case of a near UV CD where the aromatic packing was disturbed. This was again reflected back in the fluorescence spectrum where we see a reduction in intensity and also a red shift, which is indicative of unfolding. And clubbed with this, we also saw that the ANS is increasing in a concentration dependent manner. So now we are well aware that the secondary structure is distorted, tertiary packing is also altered, and then we also have an opened up hydrophobic surface area. So therefore, we know that in the presence of PEG 20,000, this particular protein is undergoing this particular protein is definitely undergoing some kind of local unfolding or global unfolding. We are yet to uncover that. Now, moving forward, we also recorded the lifetimes of this particular tryptophan residue in the presence of PEG 20,000, and we see that the longest lifetime of this particular residue, which is a 4.99, has reduced to 4.5 and 4.35, and also the shorter lifetime 1.98 is also reduced to 1.38 and 1.59. And if you see the population distribution among the longest and the shortest lifetime have completely changed. So whatever 50%, approximately 60% of the protein was in the longest lifetime distribution. So the longest lifetime holds for a tryptophan conformer which is shielded from any kind of quenching, but that has changed in the presence of PEG 100 mg per ml. You can see that that population has completely gone down to 0.08 and 0.05. So there is only one predominant conformer in the presence of PEG, and which is again relating back to the redshift that is seen in the static fluorescence. Moving forward, we did thermal denaturation studies on the upper form of the protein, and then we see that the upper form of the protein reduces in the TM by approximately 2 degrees or so. And this was done on a CD spectrophotometer. And we also see that the other thermodynamic parameters which are there, like delta H and T delta S, they also reduce. So a combined reduction of the TM along with delta H actually holds or actually represents a scenario where you have direct PEG protein contacts. So to assess these PEG protein contacts further, we recorded a TROSY spectrum, and we found, and as we see over here, we are seeing some significant chemical shift perturbations. So these chemical shift perturbations, which are now loaded in the panel next to the entire spectrum, are very much evident that these are the residues the from where PEG is trying to interact with the protein and trying to distort the entire structure of the protein and giving rise to a non-native form. So the interesting fact over here is that we also try to take a look at the intensities. So when we take a look at the intensities, so the peaks which definitely have chemical shift perturbations are all showing are all showing some or the other change in the intensity. So when you see that there is a change in intensity, either the peak is getting more intense or the peak is getting less intense, we can think of it as the protein is tumbling, which is clubbed with the which is clubbed with PEG right now. So that's one way to look at it, and therefore we can also say that the PEG protein interactions are transient in nature. When we map these things back onto the secondary structure of the protein, we see that most of the residues fall at the terminal ends of the secondary structures that are available. So this particular protein is getting affected in such a way that the terminal residues are getting affected. So the fraying of the secondary structure that generally happens because of the dynamic nature of the protein is getting affected in the presence of PEG. And we can also see that the alpha helix 4 is the most affected peak, most affected secondary structure in this particular protein. And to our surprise, alpha helix 4 is also the area where protein protein interactions take place inside the cell. So Ki interacts with Ki A, Ki Z, and FLIM via alpha helix 4, and that is what is getting affected majorly in the presence of a macromolecular crowder. So therefore, we can understand that when we are trying to, and the intensities are also widespread in a similar manner. And intensity changes are much more seen in the alpha helix 5. So the ensuing changes that are happening after the chemical shift perturbation is also visible in the following secondary structure element, which is alpha helix 5. So summary of the effect of PEG is that there are a lot of CSPs that are observed and the intensities are also changing. That means there are direct PEG protein interactions, and due to this PEG protein interaction, the protein is trying to adapt a conformation which is slightly different than its native state. And when we try to diagnose the magnesium binding ability of this particular protein, to our surprise, in the presence of 200 mg per ml, metal did not bind to the protein at all. That means all the interactions that are taking place in the upper form of the protein with PEG are distorting the protein to such an extent that the metal is not able to bind, or the metal binding site is in its non-native form. So that was the case with PEG. Moving forward, when we do the same kind of experiments with Fecol 70, which is a carbohydrate based Crowder. This is the paper that also got nominated for this Best Researcher Award. So from here moving forward, you will try to see that this is the particular thing that we are trying to. So the second researcher contrarily increases in the presence of Fecol. Tertiary packing is also slightly disturbed. Fluorescence intensity decreases, but there are no shifts. Now, this decrease in the fluorescence intensity can also be explained because the dielectric constant of the medium is changing upon addition of upon addition of Fecol. So, therefore, the reduction does not mean anything to us right now. ANS is also showing up only at 300 mg per ml, which are the concentrations that are prone to aggregation. So, ideally at 200 mg per ml, there is nothing, there is no aggregation or there is no opening of the structure that is seen at all. Now, when we do the stability of the apoprotein, we see that it is increased slightly by about 2 degrees or so, which is well within the range and does not make much of a difference. So we went ahead and recorded an HSQC spectra in the presence and absence of Fecol. And then we see that in the presence and absence of Fecol, there are no chemical shift perturbations observed whatsoever. And there are slight changes to the intensities of the peaks, but that can be attributed to the viscosity effect of this entire thing. Now, moving forward, we studied the metal binding ability of this particular protein, and we see that the metal bound kiwi is coming to a point where the stability is not all the way to the apoprotein, but it is not as high as the holoprotein. It is lying somewhere in between, and this can only be explained when we do the HSQC spectrum of this particular protein, of holoprotein, in the presence of Fecol as well. There are certain secondary structure changes that were seen. There was a change in intensity, and certain peaks for the holoprotein have shown a specific change. So this specific change that is seen only in the presence of Fecol for holoprotein stems the idea that the apo-holo equilibrium that is established in the presence of 10 millimolar magnesium also, that apoholo equilibrium is getting distorted completely. And this NMR experiment is an specific example of this particular kind of thing that we are trying to see over here. And relatively integrated peaks, and we are seeing that in the presence of Fecol. So basically, the apoholo equilibrium is what is getting distorted completely. So there are a couple of more experiments that I had tried to do, and I think the information is not available in this presentation right now. But I would like to point out that we have also done the thermal denaturation experiments in the presence of sucrose and glucose, and there we see that the holo proteins stability is increasing beyond the protein in dilute buffer condition. So, therefore, in the presence of Fecol, it's the macromolecular crowding or the macromolecular nature of the Fecol that is actually reducing the thermal stability of the protein by distorting the apoholo equilibrium and selectively destabilizing the hollow part of the protein. So the conclusions from this study are that PEG affects the structure in a manner that precludes magnesium binding. The modulation of the effect originated at the ends of the helices and beta sheets. The effect of PEG is dominantly enthalpic in nature. In Fecol alone, there is a fine balance between entropic and stabilizing and enthalpic destabilizing effect. Hence, no change in the structure or stability. Metal bound conformation is no more prone to crowding effect, as the balance is now disposed towards destabilization, and this destabilization is coming because of apoholo equilibrium being distorted. I had also attempted to do some MD simulations, but and I've tried to do some PCA. This is a topic for some other time. So the prospects of the study are listed here, and the difficulties are that recording NMR in macromolecular crowding agents is not that easy. So whatever we have done is very, very standardized and done with a lot of care. And some future work that I was planning is some in-cell NMR spectroscopy that I have done here. So that'll be all from my end. Thank you so much. And this entire presentation is dedicated to my late professor. He's no more right now. So this is a tribute to him and his work. Thank you.